all. So now in this class we have to discuss about the process called microsporogenesis. So microsporogenesis, it means a production of microspores or pollen from microspore mother cell or pollen mother cell by the process called as meiosis. So by the meiosis process, the diploid microspore mother cell can produce the haploid microspores. So the tapetum is the third layer or innermost layer which helps for the nourishment of the sporogenous tissues. With that help, total 2 thecus can produce tetrathecus anther can produce the 4 microsporangia. Each microsporangia by undergoing meiosis process produces millions of microspores. At the time of the dehiscence of an anther, the wall of the anther, all these microspores are released out, and the released microspores can be carried and those reached on the stigma and to proceed the process called as the pollination, from pollination to fertilization. So now we have to observe the Pleiades of those tetrathecus anthers. Each of the microsporangia, all those are in haploid condition, and those haploid conditioned cells can produce us the cells we called it as the pollen grains. Now we have to study the structure of the pollen grains. So the morphology of the pollen grains may differ in different plants. Some of the cells are appeared in size. They varies in size, shape and morphology. The morphology will be differ. Now we have to study here. So most of the pollen grains are appeared in a round spherical structure which is made up of 2 layers. The outer layer we call it as the exine and the inner layer we call it as the intine. So the exine layer is made up of sporopollenin, which will helps for preserving the pollen for several hundreds of years or several years. The inner layer is the second layer, which is made up of intine. It is made up of cellulose and pectin substances. And these pollen grains are in a two-celled condition. They shed in two-celled condition. One, it is a vegetative cell. The upper one we call it as the vegetative cell. So this is called as the vegetative cell. The outer layer we call it as the exine layer, and inner layer we call it as the intine layer. And it is made up of 2 cells. One, it is a vegetative cell. The bigger one we called it, which is having a higher food preserve and protoplasm, is called as the vegetative cell, and the vacuoles are present. And a oval shaped structure cell we call it as the generative cell. During the time of the production of the re-entry of the pollen tube or pollen grain into the pollen tube, during that time this generative cell can undergo one mitotic cell division to produce 2 gametes. Those 2 gametes, one it is fertilizes with egg cell, the other one is fertilizes with secondary nuclei or the polar nuclei. Then the double fertilization will be complete. So in here the process we called it as the structure of these pollen grains may varies in their morphology, color, size and shape. If you have to observe, they are commonly, the pollen grains are from 25 to 50 micrometers in their diameter. These pollen grains are performing a major role and having more nutrients, and enriched amount of the nutrients are present and proteins. So most of the country peoples, they are taking by orally, by collecting of the food in the form of food or nutrient supplement purpose, or the some of the peoples, those who are performing very good in athletes, those also they are consuming, but it is illegal. So the sports persons who are taking a direct supplement of this pollen for performing extra activities or the good performance, immediate performance, so it is a crime. And these pollens will causes allergies, especially the parthenium, the plant we call it as the parthenium or a carrot grass. So this parthenium or a carrot grass which causes a allergy and causes the diseases like bronchitis disease or the asthma diseases, like such type of the diseases will be causes, and those disease caused by the pollen grains. And these pollen grains can be preserved for several hundreds of years, or even in high temperature and humid conditions also can be preserved because of the presence of the sporopollenin layer or the outer sporopollenin coating, which helps for alkali and strong acids it will protects. And the pollen grains can be shed for hundreds of the years or several months or years because of the by using liquid nitrogen minus 196 degrees of liquid nitrogen. The liquid nitrogen can be helps for preserving of these pollen grains from several years, and these can be preserved just like a seed bank. And seeds how we are preserving like that, pollens also, the pollen banks can be preserved the pollen grains for next cropping time, like the cross breeding, or we call it as the cropping breeding time. During the time of the breeding they are using these stored pollen grains, and the pollen grains which are stored, which are having good quality, those good quality of the pollen grains can help for good productivity, and hybridized characters of the characters can be seen by the help of the a good pollen grains. And these pollen grains are preserving at the liquid nitrogen of minus 196 degrees, and it can be preserved by in vitro manner only, by laboratories only they are preserving, and that can be they placed in the pollen banks. In that pollen banks only we can collect the good quality of the microspores, or also we call it as the pollen grains. And those pollen grains are just like powdery like substances which will helps for to reach the stigma, and by reaching the stigma then they will start to produce the pollen tubes, and through the pollen tubes the gametes will be entered, and the entered gametes can be fuses with a female gametophyte, we call it as the egg cell, and egg cell and the secondary nucleus in case of the angiospermic plants. And angiospermic plants can performing the a double fertilization, a production of a endosperm. The endosperm isn't replied condition. Thank you.